Welcome to today's lecture on ABO subgroups, where we look beyond the four standard blood types to understand the complexities of antigen expression. While we routinely classify individuals into major groups like A or B, these categories often mask significant quantitative and qualitative variations in how antigens are constructed on the red cell membrane or these variations, known as the ABO subgroups, are phenotypes that differ primarily in the number of antigen sites expressed on the cell surface. At the molecular level, these subgroups are determined by the efficiency of inherited glycosyl transferase enzymes. For example, the A1 gene codes for a highly potent enzyme that creates roughly 1 million antigen sites per adult red cell, whereas the A2 gene produces a less efficient enzyme, resulting in only about 250,000 sites. Together, A1 and A2 account for 99% of all Group A individuals, yet they remain distinct because up to 8% of A2 individuals will develop an anti-A1 antibody that can cause discrepancies during laboratory testing. Beyond these common types, we will explore weak subgroups such as A3, AX, and AL, which collectively occur in less than 1% of the population. These rare phenotypes often present with weak or missing reactions in forward grouping, requiring specialized techniques like adsorption and elution or the use of lectins, such as your deleterious biflorus, to identify them accurately. Understanding these subgroups is not merely an academic exercise, it is a matter of transfusion safety. If a weak A donor is misidentified as Group O, their red cells could be destroyed by a recipient's potent anti-A B antibodies, leading to a dangerous transfusion reaction. In this session, we will break down the biochemical pathways, the genetic mutations involved, and the systematic laboratory approach required to resolve the ABO discrepancies these subgroups create. We have two major subgroups. We have the A and the B subgroups. But let's talk about first about A subgroups. So the two major subgroups of your A subgroups are A1 and A2. So the A1 and A2 genes are the primary alleles responsible for the two most common subgroups of Group A, which together account for approximately 99% of all Group A individuals. While both genes instruct the body to build A antigens on red cells, they differ significantly in their efficiency and the complexity of the structures they create. Both genes are located on chromosome 9 and code for the production of a specific enzyme, which is alpha 3N acetylgalactosaminil transferase. This enzyme's job is to attach the immunodominant sugar and acetyl D-galactosamine to the H antigen foundation. So let's talk about more about the A1 gene. So the A1 allele is highly efficient. It produces a high concentration of the transferase enzyme, which acts effectively on almost all available H antigen foundations, including both simple, linear, and complex branch sugar chains. While the A2 gene, on the other hand, the A2 allele is characterized by specific mutations, including a single base deletion, which results in a less efficient enzyme. So this enzyme is less effective at converting the H foundation into A antigens, particularly on complex branch sugar chains. So the different efficiencies of these genes lead to two major distinctions in the resulting phenotypes. So we have for A1, the number of antigen sites, it contains 810,000 to 1.17 million, while A2 has less as compared to A1 because the number of antigenic sites is 240,000 to 290,000. The amount of transferase enzyme, your A1 contains more of it, while A2 contains less of your transferase enzyme. And then the amount of branching of precursor substance. A1 is more branched, and then A2 is linear. So on the qualitative side, they differ in antigenic structure. So for A1, A antigen and A1 antigens are present, while on A2, only the A antigen is present. That's why it cannot recognize the A1 antigen. And for your A2B, only A antigen is present and it cannot also recognize A1 antigens. So that's why for the formation of anti-A1, both A2 and A2B will produce an antibody called as your anti-A1. So let's talk about the reaction of your A1 and A2 cells. So let's say for instance, you are going to use an anti-A reagent. So both A1 and A2 cells will react strongly, about 3 plus to 4 plus, grading with a standard commercial anti-sera A. For anti-A1 lectin, which is from your Dolichos by Florus, so this reagent will clump your A1 cells but fails to react with your A2 cells. For anti-H lectin, which is coming from the Ulex europaeus plant, so because A2 cells have much more unconverted H foundation, they react much more strongly with anti-H than your A1 cells do. So an individual inherits the A1 gene from at least one parent. Let's say we had the different genotypes, A1, A1, and then A1, A2, or A1O. So their cells will have the maximum number of A antigens and will clump with anti-A1 lectin. So if an individual inherits the A2 gene and lacks the A1 gene, so he or she will have the following genotypes. So A2 and A2 or A2O. So their cells have fewer A antigens and will not clamp with anti-A1 lectin. So because the A1 antigen is qualitatively more complex, some A2 individuals about 1 to 8% and A2B individuals about 22 to 35% recognize the A1 structure as foreign and produce an antibody called your anti-A1. So this can cause an ABO discrepancy in the laboratory where the person's forward type is A, but their serum unexpectedly reacts with reagent A1 cells in the reverse typing. So ABO subgroups are different versions of the group A or group B blood types that vary primarily in the number of antigen sites present on the red blood cell surface. So while most people are simply typed as group A, this category is actually a spectrum ranging from very strong antigen expression to nearly undetectable levels. So approximately 99% of all group A individuals fall into one of these two categories. So we have A1, 80% of group A, so which is the most common subgroup. The A1 gene will produce a very, very potent enzyme that creates a high concentration of A1 antigens on both complex, branched, and simple linear sugar chains. For A2, which is about 20% of group A, is the second most common subgroup. So the A2 gene produces a less efficient enzyme, resulting in significantly fewer A antigens that are located only on simple linear chains. So because the A2 phenotype is qualitatively different between 1 to 8% of A2 individuals and up to 35% of A2B individuals will produce an antibody called an anti-A1, as what I have mentioned previously. While often a harmless cold reacting antibody, it can cause confusing results or the so-called discrepancies during routine blood typing. So we also have the so-called weak subgroups of A. So these subgroups are extremely rare, occurring in less than 1% of the population, and they are usually discovered when a patient's forward and reverse typing results do not match. So the weak subgroups are A3, which is the mixed field subgroup. So it's famous for its mixed field agglutination pattern. So wherein in a test tube, you will see small clumps of red cells floating in a sea of unagglutinated cells. So we also have AX. So these red cells typically do not react with the standard anti-A reagent, but will show a reaction when tested with anti-AB. Individuals with this subgroup almost always have anti-A1 in their serum. We also have AL. This is one of the weakest subgroups, so the A antigen is so sparse that it cannot be detected by standard clamping tests at all. Scientists must use a special adsorption technique, basically washing the red cells to catch and then release any tiny amounts of antibody to prove the A antigen is actually there. So how are we going to distinguish subgroups in the laboratory? So we medical technologists use specialized proteins called lectins, which are seed extracts in order to identify these subgroups. I have already mentioned in the previous talk that I have that anti-A1 lectin is extracted from the Dolichos bifloro. So this reagent will clump your A1 cells but will not clump A2 or weaker cells. We also have anti-H lectin, which is extracted from your Urlex europaeus. So this reacts with the H antigen foundation because weak subgroups like A2 or AX use up very little H antigen in order to make A antigens. They have lots of leftover H foundation and therefore they react very strongly with the anti-H lectin. So I have here a table. So kindly familiarize this one, everyone. So identifying these subgroups is very critical for blood donor safety. So if a donor with a very weak subgroup like AX, it's accidentally labeled as group O. So their blood could be given to a group O recipient. The recipient's potent anti-A and anti-B antibodies would then attack the donor's weak A cells, potentially causing a dangerous transfusion reaction. This diagram here shows the comparison of your A1 and A2 red cells. So the A1 and A2 phenotypes are the two major subgroups of blood group A. So together accounting for 99% of all group A individuals. While both are classified as a type A in the routine testing, they differ in how efficiently they build A antigens on the red blood cell surface. So both phenotypes are created by the same type of enzyme, which is your alpha-3 acetyl galactosamine transferase. But the potency of the version inherited is different. Again, for the A1 phenotype, the A1 gene codes for a highly efficient enzyme that aggressively converts the foundation, which is your H substance, into A antigens. While for A2 phenotype, the A2 gene has specific mutation, could be a single base deletion that results in a significantly less efficient enzyme. So the primary difference of your A1 and A2 is the number of antigen sites, as what I have already mentioned from the previous slide that we have. So A1 individuals have a very high concentration ranging from 810,000 to 170,000 sites per cell. A2 individuals have far fewer, roughly 240,000 to 290,000 sites per red cell. So beyond just the number of sites, the complexity of the sugar chains also differs. So for A1, antigens are built on both complex branched and simple linear sugar chains, while the A2 structure antigens are built primarily on a simplified linear chains. So as you can see on our diagram presented, so because the A2 enzyme is less efficient, then A2 cells have much more unconverted H antigen, which is the foundation Okay. So the unconverted H antigen remaining on the red cell surface than A1 cells do. Okay. A2 phenotype have much more unconverted H antigen remaining on the red cell surface as compared to your A1 cells. So in a standard blood bank, these two can be very tricky to distinguish because both react strongly, usually 3 plus to 4 plus reaction with routine commercial anti-air agents. So to tell them apart, technologists use specialized tools such as your lectins, which I have already mentioned earlier, anti-A1 lectin, your dolichos biflorus. So it will really clamp your A1 cells, but not your A2 cells. As A2 cells have more H antigens than your A1 cells. And then anti-H lectin, which is extracted from your Ulex europaeus, because A2 cells have more leftover H foundation, then they react much more strongly with this reagent than your A1 cells. Because the A1 phenotype is qualitatively more complex, some A2 individuals recognize the A1 structure as foreign. So that's why anti-A1 is found in 1 to 8% of your A2 individuals and up to 35% of A2B individuals. So as what I have mentioned earlier, aside from that, anti-A1 is typically a cold reacting IgM antibody that is not clinically significant unless it reacts at body temperature, which is 37 degrees Celsius. So, uh, I'll give you an example of genetic outcomes for A1 and A2 phenotypes. So, if a person inherits an A1 gene from one parent and an O gene from the other, so they will express the A1 phenotype because A1 is dominant. So let's have another example. If a person inherits an A2 gene from one parent and an O gene from the other, they will also express the A2 phenotype. But a person with a genotype A2B is more likely to produce anti-A1 than someone with genotype A2O because the B enzyme competes even more effectively for the H foundation, so leaving the A antigens even more sparse and foreign looking to the immune system. Now, how are we going to differentiate A1 and A2 individuals? Both A1 and A2 individuals look identical in routine testing because their red blood cells react strongly, which is again 3 plus to 4 plus with standard commercial anti-A. The primary way to tell them apart is by using a specialized reagent called anti-A1 lectin, which is extracted from your dolichos by Flores. So this lectin specifically agglutinates A1 cells but does not react with your A2 cells. For example, if a person's blood type if a person's blood types as group A but shows no reaction when tested with an anti-A1 lectin, they are classified as an A2 individual. Another method of differentiation involves the H antigen, which is the foundation used to build A antigens. Because the A2 gene produces a less efficient enzyme, it leaves more foundation unmodified on the cell surface. Consequently, A2 cells react much more strongly with anti-H lectin extracted from your Ulex europaeus than A1 cells do. You can also differentiate them by looking at a patient's serum or the reverse typing because between 1% and 8% of A2 individuals and as well as 22 to 35% of A2B individuals produce an antibody called anti-A1, while A1 individuals do not. For example, a common laboratory clue for an A2 individual is a discrepancy where the forward type is group A, but the patient's serum unexpectedly reacts with reagent A1 cells. So, the biological reason for these differences is both quantitative and qualitative. So, for quantitative, could be A1 red cells have a high density of antigens, approximately 1 million sites per cell, while A2 cells have significantly fewer sites, no, approximately 250,000 sites. And in terms of complexity, A1 antigens are built on both complex branch and linear sugar chains, whereas A2 antigens are primarily found on a simple linear chains. So again, both A1 and A2 cells will react with the commercial anti-Sera A. So these are the two methods that could really differentiate them. So you can distinguish both serologically with A-lectin. Again, the Dolichos biflorus is extracted from the seeds of the plant and they possess an anti-A1 specificity. So adsorption, it's another method. So it's a technique that uses your red cells with known antigens to remove specific antibodies from a solution. So this process is used to create a specialized reagent from human serum, typically from group B or group O individuals that contains both anti-A and anti-A1. So how are we going to do this? You're going to incubate human anti-A serum with your A2 red cells. And then the A2 cells will absorb the anti-A component of the serum because they possess the A antigen. However, since A2 cells lack the specific A1 determinant, the anti-A1 component remains in the liquid portion or in the serum. Once the A2 cells are removed, the remaining liquid is called absorbed anti-A1. So these reagent can then be used to test unknown group A cells. If it causes agglutination, the individual is A1. If it does not, they are A2. So again, lectins are plant seed extracts that behave like antibodies by binding to specific carbohydrate determinants on red blood cells, causing agglutination. So next we have your weak A subgroups. So weak A subgroups have various characteristics, which includes reduced expression of A antigens. Decrease of antigens per cell results in weak or no agglutination when tested with common anti-Sera A. And then due to the inheritance of rare alleles at the ABO locus, that's why you have weak A subgroups, and then it occurs at less than 1% frequency. And then occasionally, weak subgroups of A may present practical problems. Let's say, for instance, an AX donor was mistyped as type O and was transfused to a group O patient. So this is potentially dangerous because group O possesses anti-A and anti-B, which agglutinates and lyses the AX RBCs, causing rapid intravascular hemolysis. Again, as what I have mentioned earlier during our discussion, that weak A subgroups are rare variations of the group A blood type that occur in less than 1% of the population. So unlike common A1 and A2 types, these subgroups have a significantly reduced number of A antigens on their red blood cells, which leads to weak or even missing reactions during routine blood typing. So these subgroups are caused by the inheritance of rare alleles at the ABO locus on chromosome 9. These rare genes produce a less efficient enzyme or glycosyl transferase that struggles to attach the A sugar to the H antigen foundation. So because so few A antigens are created, the cells are left with an abundance of unconverted H antigens, often as many as group O cells. So upon testing your weak A subgroups, they will produce a weak forward typing result. So they will show 1 plus or no reaction with the standard commercial anti-A reagents. But upon testing it with your anti-H lectin, your weak A subgroups will really react very strongly, usually 3 plus to 4 plus. And then There are also some individuals that will produce an anti-A1 antibody in their serum, which can cause a mismatch in the reverse type. So if the person is a secretor, A antigens may still be detectable in their saliva even if they are hard to find on the RBCs. So the weak subgroups are classified in this table. So it's based on their reactivity with human anti-A and anti-AB, and as well as the site in the RBC. So let me talk about first your A3, which is the mixed field subgroup. This is the most defining feature of A3. Um, I'm talking about mixed field agglutination, so that's the most defining feature. So when you're going to test it with anti-A, you will see small solid clumps of cells surrounded by a background of unagglutinated cells, or meaning those are just free cells floating around. So this is a natural genetic expression and it not a result of a recent transfusion. That's for A3. Now let's proceed to the AX. AX is sometimes known as the anti-AB responder because AX cells usually do not react with the standard anti-A. However, they will show a clear reaction, which is weak, 1 plus to 2 plus with anti-AB. So it is a special reagent that is better at detecting weak A expressions. So these individuals almost always have anti-A1 in their serum. Next, we have your A subscript L. So sometimes this is referred to as the invisible subgroup. AL is so weak that it cannot be detected by standard clamping tests at all. Usually it reacts zero with anti-A and anti-AB. So to prove the to prove that A antigen exists, we medtechs must use adsorption. So we are going to soak the cells in anti-A and then wash away the excess and then pop the antibody off to see if it was captured by any hidden A antigens. So for our subscript M, it does not usually react with anti-A, but the person will have normal levels of A substance in their saliva. For A subscript N, it shows a very weak mixed field uh, reaction where less than 10% of the cells actually clump. And for AY also, it's actually similar to AL. It shows no reaction with anti-A and requires adsorption, but it has trace amounts of A substance in the saliva. So how are we going to identify the weak A subgroups? So for A3, you have a mixed field pattern of agglutination with anti-A and as well as with anti-AB. So when we talk about mixed field, um, again, these are small agglutinates within predominantly unagglutinated cells. So why does it matter in order to identify these weak A subgroups? So it is really critical for blood donor safety. So if a weak A donor is mislabeled as group O, their blood could be given to a group O recipient. So the recipient's natural anti-A, B antibodies would then attack the donor's weak A cells, causing decreased red cell survival or a transfusion reaction. So in contrast, if a weak A recipient is typed as group O, they will simply receive group O blood, which is safe for them. So to identify weak A subgroups, again, which occurs in less than 1% of the population, we should follow a systematic process of serologic testing to resolve ABO discrepancies. So these subgroups are characterized by a significantly reduced number of A antigen sites on the red cells membrane, which will lead to a weak or missing reactions in the routine testing. So how are we going to uh, resolve. So the first thing is to recognize the ABO discrepancy. Identification usually begins with forward grouping or the forward typing. Usually testing RBCs with anti-A shows a weak or 1 plus or negative reaction, but the reverse grouping in which you're going to test the serum does not match the expected pattern for group O. For example, the RBCs may react as group O, but the serum contains only anti-B, suggesting the person is actually group A. So to detect sparse antigens, we should use more sensitive reagents and techniques. So that's why we have the so-called anti-AB reagent, not anti-A alone and not anti-B alone, but the combined anti-AB reagent. So while monoclonal anti-A has become very sensitive, anti-AB, especially human polyclonal or specific monoclonal blends, is historically used because it is often more effective at detecting weak subgroups like your A subscript X. So agglutination can be enhanced by incubating the RBCs and antisera at room temperature for 30 minutes or at 4 degrees Celsius for 15 to 30 minutes in order to increase the antibody association. And aside from that, you can also use lectins, in which again, lectins are plant extracts used to differentiate your weak A subgroups. So for anti-A1 lectin, your weak A subgroups will react negatively with this lectin, confirming they are not A1. So for anti-H lectin, because weak A subgroups convert very little H antigen into A antigen, they have an abundance of H foundation. So these cells will react very strongly, which is 3 plus to 4 plus grading with anti-H similar to group O cells. So for the weakest subgroups such as your A subscript L, A subscript M, or A subscript Y, that show no agglutination even with the combined or polyclonal anti-AB, we should use adsorption or elution, can be elution. So in which you're going to incubate the RBCs with anti-A in order to allow any tiny amounts of the antigen to catch the antibody. And then after that, the antibody is then chemically or can be physically removed, which is the process of elution, and then you're going to test it. If the eluate reacts with the group A reagent cells, it proves the A antigen was present on the original cells. So in terms of saliva studies or the secretor status of the weak A subgroup, if the individual is a secretor, meaning it inherits the big S small e gene, so their saliva can be tested for soluble A and B. Soluble A and H substances. So subgroups like AM will show normal amounts of A substance in their saliva, even if RBC's reactions are nearly invisible. Others like A subscript L or A subscript X will only show H substance in their saliva. Again, accurate identification is vital for donor safety. So if an AX donor is mislabeled as group O, their blood could be transfused to a group O recipient, and then the recipient's naturally occurring anti-AB would then attack the donor cells, potentially causing a hemolytic transfusion reaction. So this diagram shows the ranking of A subgroups um, from with the most A antigens to the fewest A antigens. So the ranking of A subgroups is determined by the antigen density, which refers to the total number of antigen sites present on the surface of a single RBC. So this density is a direct result of how efficiently an individual's inherited glycosyltransferase enzyme converts the foundation H antigen into A antigens. So based on the adult RBC antigen counts, the subgroups are ranked from highest to lowest concentration as A1. It has the most common subgroup. It's the most common subgroup. It possesses a high concentration of A antigens built on both complex branch and linear sugar chains. Next, second on the ranking, we have A2, the second most common type. usually 20% of group A individuals. So it has significantly fewer antigen copies than A1, which are located only on a simple linear chain. A3, it's a rare subgroup, less than 1%, known for a mixed field reaction, where small clumps of your cells are seen among many unagglutinated cells in laboratory testing. Then we also have A subscript X. So these cells typically do not react with standard anti-A reagents and often require a specialized anti-AB reagent for detection. And lastly, the fewest A antigen of your A subgroups is your A subscript um, L. No, it has an extremely weak subgroup that shows even less reactivity than your AX, with only about 10% of the cells showing tiny clumps. Next, I have here a table summarizing the different reactions of your um, weak A subgroups, and then please do memorize this one. Next, we have B subgroups, which are rare variations of the group B blood type that are characterized by a significantly reduced number of B antigen sites on the red blood cell surface. While they follow the same general principles as A subgroups, they are much rarer in the population. So, like A subgroups, these are caused by the inheritance of the alternate alleles at the ABO locus on chromosome 9. So these rare genes produce a version of the beta transferase enzyme that is less efficient at attaching the B sugar or the D galactose to the H antigen foundation. Because the enzyme is less effective, the RBCs end up with very few B antigens and a high amount of unconverted H antigen. Now let's talk about the different B subgroups. So we have the B3. B3, it's the mixed field subgroup, just like with your A3 subgroup. Its defining feature is a mixed field agglutination pattern when tested with your anti-B reagents. This means you will see small clumps of cells surrounded by many unagglutinated or free cells. These individuals do not typically produce anti-B in their serum and have normal levels of B substance in their saliva if they are secretors. Again, B3, it's the most frequent weak B phenotype. Now let's talk about the B subscript X. So sometimes it can be referred to as the anti-B producer. So your B subscript X cells typically show very weak agglutination with anti-B and anti-AB reagents. A unique identifying feature is that these individuals often have a weakly reactive anti-B in their own serum. Despite technically being group B, if they are secretors, their saliva contains large amounts of H substance but usually no B substance. We have the B subscript M. So in this subgroup, the A and B genes are present but the antigens are almost completely missing from the RBCs. So reacting zero or only weakly with anti-B, anti-sera. However, if the person is a secretor, they will have normal amounts of B substance in their saliva. So this subgroup is more commonly reported in Japan and is thought to be caused by a modifier gene that prevents the B antigen from being expressed on the blood cells specifically. So we also have the B subscript L. This is sometimes can be called as the invisible subgroup. So this is an extremely rare phenotype where the B antigen is so sparse it cannot be detected by standard clumping or agglutination tests. To identify it, so we should use adsorption and can be elution, a process where the cells are soaked into your anti-B to catch any tiny amounts of antigen and then washed in order to prove that they were there. So these individuals may have a weak anti-B in their serum and do not secrete B substance in the saliva. Okay, so in order to tell the B subgroups apart, we should look for weak or mixed field clamping with anti-B. And then we're going to check if the person has an expected anti-B in their serum, which is common in your B subscript X and B subscript L. So if the person has the secretory gene, testing their saliva for soluble B substance is a major clue for identifying your BM or your B3. So in order to find the hidden antigens in the weakest types like B subscript L, then adsorption is really, really required. So just like with the weak A subgroups, identifying B subgroups is vital for transfusion safety. If a weak B donor is accidentally labeled as group O, their blood could be given to a group O patient. Then the patient's naturally occurring anti-B would then attack the donor cells, potentially causing a hemolytic transfusion reaction or a decreased survival of the transfused cells. So failure to detect a weak subgroup could have serious consequence. So for the recipient, can be classified as group O and then can be also transfused with group O red cells. For the donor, there could be error in labeling blood with O due to undetected weak subgroup, which can cause also to a decreased survival of the transfused red cells in your group O recipient. Now, this just shows the variation of H antigen concentrations in different ABO phenotypes. So the H antigen serves as the mandatory acceptor molecule as a review, no, or a foundation upon which your A and B antigens are built. The variation in H antigen concentrations across different blood types occurs because the enzymes produced by the A and B genes convert this H building block into A and B antigens. Because these enzymes vary in their efficiency, the amount of unconverted H antigens left on the red blood cell surface differ significantly between phenotypes. So based on the efficiency of these biological builders, so the blood types are ranked from the most H antigen to the least H antigens as follows. So group O, it has the most H antigens because individuals with group O possess non-functional O genes that do not produce an active enzyme. So as a result, the H foundation remains completely unmodified. So leaving group O cells with the highest concentration of H antigen. For the group A2, it produces a less efficient enzyme than the A1 gene. This lazy enzyme only converts some of the H foundation into A antigens, leaving a significant amount of H detectable antigens on the cell surface. For the B gene, it produces an enzyme that is moderately efficient at converting the H foundation. While it uses more H than the A2 enzyme, it still leaves more H behind than the potent A1 enzyme. Next is our A2B. So this phenotype inherits a less efficient A2 enzyme alongside a B enzyme. So the combination results in more conversion than a solo B or A2 type, but still leaves more leftover H than your A1 phenotypes. Now, next is the A1. The A1 gene is a highly potent builder. So its enzyme aggressively converts almost all the available H foundation into A antigens. So leaving very little unconverted H behind. So for the A1B, it has the least or the fewest H antigens. So because both the potent A1 enzyme and the efficient B enzyme are working simultaneously to use up, and the H foundation, the cell is left with the smallest amount of leftover H antigen. So in the laboratory, this variation is easily seen when testing with anti-H lectin or Ulex europaeus. Group O and A2 cells react strongly, usually 3 plus to 4 plus with anti-H because they have so much foundation available. Conversely, A1 and A1B cells react very weakly or not at all because their H foundation is almost entirely converted. So next we have here your lectins. So lectins are specialized proteins, most commonly extracted from plant seeds, though they can also be found in bacteria and mammals. And in the blood bank, they are used as highly specific reagents to identify certain blood group antigens. So while prolectins, on the other hand, they are specific antibodies derived from snails. So the key characteristics of your lectins, unlike a traditional anti-sera, lectins are not antibodies, but they are not also produced by an immune system. So they works by your lectins work by specifically binding to the carbohydrate portion or the immunodominant sugar of a certain red cell antigen. So when a lectin binds to its target sugar on the RBC membrane, it causes the cells to agglutinate or clamp together, providing a visible positive result. So lectins are excellent alternatives to human anti-sera because they are highly specific and do not contain contaminating antibodies that might be found in human-derived products. So they are particularly useful when a patient has a subgroup like A2 that has developed an antibody which is anti-A1 against the more common A1 antigen. So we have here the common lectins used in blood banking. So we have anti-A1 lectin, Dolichos biflorus. This is the most important lectin for routine work. It specifically agglutinates the A1 and A1B red cells, but does not react with A2 cells. So it is used to distinguish between A1 and A2 phenotypes with resolving ABO discrepancies. Next, we have anti-H-lectin. Anti-H-lectin, it's your Ulix europaeus. So this lectin is specific for the H antigen foundation. So it reacts strongest with cells that have the most unconverted H antigens, such as group O or weak A subgroups. We also have anti B lectin, which is extracted from your Banderia simplicifolia. So it will react only to your true B cells. Next, aside from the mentioned anti-A or anti-A1, anti-B, and anti-H lectins, we also have anti-N lectin, which is from Visea graminea. So this lectin has specificity for the N antigen in the M and S blood group system. We also have an anti-M lectin, which is extracted from your Iberis amara. So this lectin is specific for the M antigen. Next, we have here the different specificity and the source of your specific prolactin, so kindly familiarize everyone. So next, we have your Bombay phenotype. Again, it was first reported by Bende in 1952 in a family living in Bombay, India, which is the source for the phenotype term. So what happened here in a patient with Bombay phenotype? There are no big H antigens and no ABO antigens. So the serum and the red cell reactions similar with group O in routine testing, but the anti-H, it's significant because it reacts at body temperature. So there are 130 reported cases with greater incidence in India. So if ABO genes are inherited, so the genes can be written as O subscript H and then superscript A, and then O subscript H superscript B if they have the B gene. Then for AB genes, so O subscript H and then superscript AB. So again, the Bombay phenotype, it's a rare genetic occurrence that significantly alters routine ABO blood typing results because these individuals lack the H antigen, which is the essential foundation for A and B antigens. So in standard laboratory testing, an individual with the Bombay phenotype will really appear to be group O. So their red cells show no agglutination when tested with the anti-A or anti-B reagents because they lack the A and B antigens. So their serum contains all of the antibodies, anti-A, anti-B, and anti-AB, just like a normal group O individual. So the critical difference between a normal group O individual and a Bombay individual lies in the H antigen. Normal group O individuals have the highest concentration of H antigens in their RBCs, while the Bombay phenotype, because they inherit the rare small h genotype, they do not produce the enzyme L-fucosyl transferase needed to create the H antigen. So without this foundation, even if they have inherited the A or the B genes, those antigens cannot be expressed. Again, Bombay individuals produce a potent anti-H antibody in their serum. This antibody has high thermal activity at 37 degrees Celsius and can activate the complement, causing intravascular hemolysis if H positive blood, like the normal group O blood, is transfused. So, to differentiate a Bombay individual from a standard group O individual, you can use specialized reagents such as your anti-H lectins, which is extracted from your Ulex europaeus. Normal group O cells will react strongly, usually 3 plus to 4 plus in grading, with this anti-H lectin, while the Bombay RBCs will not react at all, usually 0. So Bombay serum will agglutinate all standard ABO reagent cells, including group O screening cells, and then because all normal RBCs possesses the H antigen. So accurate identification of Bombay phenotype is vital for transfusion safety because a Bombay individual is compatible only with other Bombay individuals. If they are misidentified as group O and receive group O blood, they will suffer a severe, potentially fatal hemolytic transfusion reaction due to their potent anti-H. Now we have H deficient phenotypes, which are rare blood types characterized by the partial or complete absence of the H antigen, which is essential foundation or the building block required to build the A and B antigens. Because the H antigen is the mandatory acceptor molecule for ABO sugars, individuals with these phenotypes cannot express normal levels of A or B antigens on their red blood cells even if they have inherited the functional A or B genes. So these genotypes are generally divided into three categories. Category 1, we have the classic Bombay phenotype. Category 2, we have the RBCH partially deficient non-secretor. And category 3, which is the para-Bombay. Now let's proceed to the classic Bombay phenotype or the big O subscript H. So it was first discovered in Bombay, India, and then this is an extremely rare genetic occurrence where the individual lacks the H antigen entirely. So it results from the inheritance of two recessive small h genes and two recessive small se genes. So the individual cannot produce the enzyme L-fucosyl transferase. So without this enzyme, the body cannot attach the sugar L-fucose to the precursor chain to form the H antigen. So your classic Bombay phenotype, they appear to be group O because their red cells lack A and B antigens. Unlike normal group O individuals, Bombay red cells will not react with your anti-H lectin. So again, the serum of your classic Bombay phenotype, they contain naturally occurring anti-A, anti-B, and anti-AB, and a very potent anti-H. So category 1 is non-secretors, meaning they do not have A, B, or H substances found in their saliva or other body fluids. So let's talk about your category 2. So they have weak A and B antigens, but they do not have the residual H antigen. For category 3, the para-Bombay phenotype, so they are those whose red cells either completely lack the H antigen or express it in only trace amounts, but they differ from the classic Bombay because they can still produce A, B, and H substances in their secretions. So meaning the para-Bombay individuals usually have a mutated or silenced H gene or the FUT1 but possesses at least one functional secretor gene, the big S and then the small e gene. So depending on which ABO genes they inherited, they are categorized as A small h, B small h, or AB small h. So their red cells may type as group O or show very weak irregular reactions with anti-A or anti-B. And then they typically do not react with anti-H or react only very weakly. So they possess a weaker anti-H in their serum compared to the classic Bombay phenotype. Because your para-Bombay individuals have a secretor gene, so they do not they do secrete A, B, or H substances in their saliva. So identifying H deficient phenotypes is a critical matter of transfusion safety. So in routine testing, these individuals are often misidentified as group O. So because their serum contains a potent anti-H that reacts at body temperature or 37 degrees Celsius, they will suffer a severe, potentially fatal hemolytic transfusion reaction if given normal group O blood. So again, para Bombay or your Bombay phenotypes or the Bombay individuals are only compatible with only with the H deficient donors because transfusion management usually requires a stored autologous unit, meaning their own blood or blood from the siblings with the same phenotype or contacting rare donor registries. So we have here also the other characteristics or the other uh, things that you will need to know about the different category of our H deficient phenotype. So kindly memorize this one. And then this table shows the ABO compatibility. And ABO compatibility is the foundation of transfusion safety, ensuring that the antigens on a donor's red blood cells do not react with the antibodies in a recipient's plasma. So if incompatible blood is transfused, the recipient's antibodies can cause the immediate destruction or lysis of the donor's red cells, leading to a potentially fatal acute hemolytic transfusion reaction. So compatibility requirements differ depending on whether you are transfusing red cells or plasma or the whole blood. So for RBC transfusions, the goal is to ensure the recipient's antibodies do not attack the donor's antigens. So if you can still recall the rule, a recipient can receive donor cells as long as they do not have the corresponding antibody in their plasma. So group O RBCs lack both A and B antigens, meaning they can be safely given to any recipient because there are no antigens for the recipient's antibodies to attack. Group AB individuals lack both anti-A and anti-B antibodies in their plasma, allowing them to safely receive RBCs of any ABO type. For plasma compatibility, it is the exact reverse of RBC compatibility. Here, the goal is to ensure that the donor's antibodies, which are found in the plasma, do not attack the recipient's red blood cells. So donor plasma must be compatible with the ABO antigens on the recipient's red cells. So group AB plasma contains no anti-A or anti-B antibodies, making it safe for any recipient. Because group O red cells have no A or B antigens, they cannot be attacked by any ABO antibodies, making group O individuals the universal recipients for plasma. So in emergencies where group AB plasma is unavailable, some facilities use group A thawed plasma for trauma patients of unknown blood type. For whole blood transfusions, it contains both the donor's red cells or the antigens and the donor's plasma or antibodies. Because both components are present, the requirements are much stricter. So whole blood must be ABO identical to the recipient. For example, a group A recipient must receive group A whole blood. They cannot receive group O whole blood because the anti-A and anti-B in the donor's O plasma would attack the recipient's A cells. Because of these rigid requirements, whole blood has limited use in modern medicine and is mostly reserved for massive hemorrhages or specific autologous situations. And with that, everyone, I would like to conclude our discussion on ABO subgroups. So I hope that you have learned something from this video.